cancer is a genetic disease, so each cancer has different genetic makeup. For example, lung cancer is not the same in every lung cancer.
It's important for patients to know the genomic profile of their tumor characteristic because it gives us an idea as to what went wrong in the DNA or the system that caused this progression or development of the disease.
This information can lead us to the point that we can find targeted therapy or immunotherapy or combination of these novel therapy that could help patients.
There are certain terms of how I select the patients. One is a very complicated case, meaning to say that the data, for example, of standard regimen is not as good as you expected. Another consideration would be for patients who are already on their 4th, 5th, or 6th line of treatment, and we want to know exactly what else can we give them. So these patients would go through genetic profiling or genomic profiling in that sense.
Many cases of the cancer patient that already gone through standard treatment and we are kind of stuck. We got this information from the case that seemed to be dead end.
When we know about this particular alteration, we can use this particular drug to the patient. So that's why we need to try to find these alterations so we can treat the cancer or treat the patient with more effective and less toxic.
The NGS platform has really improved the technology and the way we treat our patients nowadays.
We need to have a technology to detect all of these abnormalities at the genetic level. So nowadays we have a technology that we can do the massive sequencing of the genes, that we can be able to find many alterations in a very short period of time.
Comprehensive genetic profiling seems to be more useful than regular PCR based of the single gene that we usually use before. This information is very important if we know. Many times we don't know about this and we treat our patient and we don't get good results. If we look back and come back for the comprehensive NGS, then we know that actually we are missing something from the beginning. I believe it's benefit every stage of cancer or some kind of the cancer that the guideline clearly show that understanding their genetic background upfront, guiding us to the better direction of treatment such as non-small cell lung cancer.
Genomic profiling is very useful, I should say. And in fact, I have been advocating this method to be used upfront for patients. The reason being is that it's better to qualify from the onset what kind of treatment we're supposed to give them, what's the driver mutation that's causing all these issues. And most importantly, it will provide us options, not just for the first line, but for second line, third line, and so on.
Cancer that has a many targeted drugs like lung cancer, you can do it up front. And some other cancer when it's resistant to the conventional treatment, or some rare cancer that you might find something that really helpful to the patient compared to the conventional drug.
In terms of cost effectiveness, data has shown that it's very important and practical for patients to have this done. The reason being is that if you do this on a later stage when the patient is already on multiple lines of treatment, sometimes you find the management that was given, they're resistant to it in the beginning or they're not appropriate candidate for that treatment from the onset. And then they already spend so much of their money and time and effort and side effects in terms of treatment as well. And only to find out that after you do the molecular profiling that they don't respond to it or they're not even candidates or they're not appropriate for that particular form of treatment. So at the end of the day, if you know from the onset what you're looking for and you know how to treat them from the onset, then it becomes very cost efficient rather than you give multiple lines of treatment, do the profiling, and then only to find out that the rest of the treatment were not as effective or efficient than what you hope and expect to be.
First of all, most of the patients doesn't know about this technology and they don't understand it. The important part is you have to explain and convince them the usefulness of it to allow us to do the test on their sample. I think the doctor-patient relationship, the trust on doctor is very important. Of course, most of patients who come to us, some of them they have very limited knowledge in science, in medicine. But once they trust the doctor, and then we also have enough time to go through with them or their family who are able to understand better, then they come up with agreement to do the test, and many times it's very helpful on them.
They might have a little bit knowledge about the molecular basis of the cancers. And because of this field growing every day, so nowadays the molecular information for the cancers is expanding a lot, and there are many targeted treatments available. So that's why we need to start to educate our teams. And then we start to have a clinical correlations to incorporate the treatment decisions into the tumor boards, and also with they contribute from the basic scientists, from the vendors to join and discuss and educate about the findings and the technologies. Make it easier and more practical and more applicable to the treatment or decision making to treat the patient.
I always try to send big panel if they can afford, because we can get some information that we didn't expect to see. We explore into the pathway and some of the information very helpful. But of course, if you want to know or do something more than standard guideline, you need to do bigger profile.
The consideration between a small panel or a large panel will depend on the tumor characteristic or the biology of the patient, or where at the stage they are in terms of treatment. So for example, if you have a very strong data in terms of the profile of the patient based on immunohistochemistry staining, based on routine histology, then you only need a certain few details in the molecular profiling that you want to order and look into. If you don't have a clear picture as to what it is in terms of the tumor characteristic, then it's better to have a bigger panel rather than a smaller panel. It will give you much better option in terms of the treatment landscape for the patient.
In short, I think nowadays we have a big advance in the molecular technologies. So we should incorporate molecular approach in our daily practice.
Well, it's almost like a breakthrough. It's a tool for us to be able to understand our patient inside out.
I would recommend that they should do it from the onset of the disease process, so that we can better characterize their disease and then give them proper and more targeted and personalized form of treatment. So it's very practical and logical to do it from the onset.